All right, today we are diving deep into one of the biggest scientific adventures in history. We're talking about the massive, worldwide effort to read our own instruction manual, the entire sequence of human DNA. This is the story of the Human Genome Project, and it all kicks off with one single, incredibly tricky question. You know, before this whole thing started, this was one of the great mysteries in biology. Scientists knew that genes were the blueprints for, well, everything that makes us us. But how many blueprints does a person actually need? We're pretty complex creatures, so you'd think the number has to be gigantic, right? And that's exactly what they thought. The best guess, the consensus from the world's top geneticists, was that we had to have somewhere around 100,000 genes. I mean, it just makes sense. A complex organism must have a really, really long parts list. Well, it turns out, not quite. When the final count came in, the entire scientific community was just floored. The real number of protein-coding genes is only about 20,500. Yeah, that's less than a quarter of what everyone expected. To put that in perspective, that's roughly the same number of genes as a mouse. This was the first huge shock from the project. So this huge miscalculation, it was a pretty humbling moment for science. And it sent us on a whole new quest to figure out what our DNA was really telling us. Because clearly, the story of what makes us human is way more complicated than just counting up the genes. So, how in the world did we get this number in the first place? I mean, sequencing 3 billion letters of genetic code is a truly monumental task. And it actually sparked this incredible competition. Two major teams went head-to-head, turning this scientific mission into a flat-out, dramatic race to the finish line. In one corner, you had the Public Project. This was a massive international team funded by governments, and their whole philosophy was collaboration and making every bit of data free for anyone in the world to use. And in the other corner, you had Celera Genomics, a private company led by the brilliant and very ambitious scientist J. Craig Venter. Their goal was speed, competition, and maybe even patenting their discoveries. But this race wasn't just about money or ideals. The two teams used fundamentally different strategies to actually read the genome. This was a high-stakes scientific bet on which method would actually work for a project this enormous. The public team used a very methodical map-first approach. Think of it like this. You have a giant unread book. First, you create a table of contents, mapping out where every chapter begins and ends. Then you break each chapter into smaller pieces, read those pieces, and use your map to put everything back in the right order. It was slow, it was careful, but it was thorough. Celera, on the other hand, went for pure brute force speed. Their method was like taking that same book, tossing it into a shredder to create millions of tiny fragments, and then using a supercomputer to find all the overlapping words and sentences to piece the whole thing back together at once. It was audacious, risky, and completely dependent on some serious computing power. So it really came down to a trade-off. The public method, it was slower, sure, but having that map made it way less likely to make mistakes, especially in the really repetitive, tricky parts of our DNA. The private method was way faster and cheaper, but it ran the risk of messing up the final assembly, like accidentally putting a sentence from chapter 1 into chapter 10. In the end, they both kind of won. The competition pushed everyone to work faster and better, and in 2001, they jointly announced the first draft of the human genome. And with the code finally in hand, the real discovery could begin, uncovering the deepest secrets hidden in our DNA. The first thing that just blows your mind is the sheer scale of it. The finished sequence had 2.85 billion nucleotide base pairs. Those are the A's, C's, T's, and G's that make up our genetic code, just a staggering amount of information. But the real question was, what does it all do? And this is where it gets really, really weird. When they broke it all down, they found that those 20,500 genes, the actual blueprints for proteins, make up only about 1.2% of our entire genome. Just over 1%. Meanwhile, almost half of our DNA is made of these things called jumping genes. This discovery just turned our whole understanding of the genome upside down. For decades, scientists had this term for all the non-coding DNA, junk DNA. They figured it was just evolutionary leftovers. But the Human Genome Project proved that could not be more wrong. This so-called junk is actually crucial. It acts like a massive, complex switchboard that tells our 20,000 genes when to turn on, when to turn off, and how much to work. It's not junk at all, it's the operating system.
The finished sequence also gave us this profound look at our place in the tree of life. For instance, get this, our DNA is only about 1% different from a chimpanzee's, just 1%. And locked inside that tiny difference are all the secrets to what makes us uniquely human.
Okay, so beyond all these incredible scientific revelations, what does all this actually mean for us in our daily lives? How has having this genetic code actually changed the world? Well, the applications have been nothing short of revolutionary. Let's start with medicine. The impact here has just been immense. We can now diagnose thousands of diseases caused by a single gene. We can look at our DNA to see if we're at a higher risk for things like cancer or heart disease. And we can develop these amazing targeted drugs that attack a disease based on its specific genetic signature. We're moving into an age of personalized medicine, where treatments can be custom-made for your specific DNA.
Then you have forensics. Genomics has totally transformed the justice system. The ability to sequence DNA with pinpoint accuracy is an incredibly powerful tool. We can identify suspects, but just as importantly, we can exonerate people who were wrongly convicted. It helps us identify victims of disasters and, of course, confirm family relationships.
And the impact just keeps spreading. We're using genomics to rewrite human history, tracing ancient migrations across the globe by looking at DNA. It's changing agriculture, helping us create crops that can resist disease and drought. We're even engineering tiny microbes to create new biofuels. It's everywhere. But with this kind of incredible power comes an equally profound responsibility, right? Being able to read our own genetic code opens up this whole Pandora's box of complex ethical questions that, honestly, we're still figuring out today.
What's amazing is that the scientists behind the Human Genome Project saw this coming. From day one, they dedicated a real chunk of their budget to a program they called ELSI to study the ethical, legal, and social issues that they knew would come from their work.
And this forces us to ask some really tough questions, like who gets to see your genetic information? How do we make sure that employers or insurance companies can't discriminate against you based on your DNA? These aren't just sci-fi scenarios anymore. They are very real challenges we have to face.
So, the Human Genome Project gave us the book of life. It taught us how to read our own code. But now, with new technologies, we're learning how to edit that code. And that leaves us with one final, pretty awe-inspiring question to think about. Now that we can read our genetic code, what on earth are we going to choose to write?